Okay, so let's take a few minutes to go over these cell cycle checks for understanding quiz answers. If you had difficulty, it was probably in this first section where I asked you to fill in the blanks without a word bank or anything, but well, let's just see how you did. So the first objective was to describe the events that occur in the cell cycle. So 1, the cell cycle consists of sequential stages of interphase, mitosis, and cytokinesis. 2, the cell can enter a stage called G0 where it no longer divides. Next objective, explain how mitosis results in the transmission of chromosomes from one generation to the next. Well, mitosis plays a role in growth, repair, and asexual reproduction. You could have had the order of these reversed. 2, mitosis alternates with interphase in the cell cycle. And 3, mitosis occurs in a series of sequential steps called prophase, metaphase, anaphase, and telophase. The next objective is to describe the role of checkpoints in regulating the cell cycle. So there are a number of internal and external checkpoints regulating the process. Again, you can have those flipped around, but it's internal and external. And interactions between cyclins and cyclin-dependent kinases control the cell cycle. I gave you that hint there for CDKs. You just have to remember what that meant. And then the last objective, describe the effects of disruptions to the cell cycle on the cell or organism. So disruptions in the cell cycle may result in cancer, which is uncontrolled cell growth, and or programmed cell death called apoptosis. All right, now your application questions. So question 1, you're supposed to figure out basically what happens next. So the first thing you have to know is that this is anaphase. So we'll start with the correct answer. The correct answer is the formation of a cleavage furrow. So you have to recognize that that's anaphase. I know that because the chromosomes are being spread apart. We're not quite getting that pinch-in part of the cytoplasm yet. So then that means that telophase will follow. So the spindle formation and nuclear envelope breaking down, those are incorrect choices because those all happen in prophase, and that's going to happen at the beginning. And this is an animal cell, so there's no cell wall and there's no cell plate that's going to form. All right, question 2. How do the daughter cells at the end of mitosis and cytokinesis compare? Well, they're going to be the same. You're going to have the same number of chromosomes and the same amount of DNA. That's why we replicate the DNA before this mitosis begins. Number 3, Taxol's an anti-cancer drug, yada, yada, yada. Taxol must affect. Okay, let's start with the answer first. It's going to be the fibers of the mitotic spindle. They're going to disrupt microtubule formation. And remember, microtubules are what form these spindle fibers. Number 4, cytokinesis. Usually, but doesn't always follow. What would be the result? Well, you're going to get a cell that has 2 nuclei. So think about it. Everything that is going to happen has happened except at that very last stage where we divide the cytoplasm. So you just have a binucleate cell. Okay, number 5. This was probably one of the more difficult ones, and I guarantee if you missed this one, it's because you chose anaphase, but let me tell you why that's not quite correct. So we always think of prophase, then metaphase, and if you look at diagrams, it shows everything unorganized in prophase, and then boom, in metaphase, everything's miraculously lined up at the middle. What we don't really talk about is this little in-between phase known as prometaphase, and this is kind of like it's the end of pro and the beginning of meta, and so the nuclear membrane's breaking down and the spindle apparatus is forming. But remember, the function of the spindle apparatus is to get all attached to the chromosomes, help them align at the metaphase plate. So really, the drugs mentioned in number 5 are going to affect metaphase. And then finally, in number 6, if you get the go-ahead signal, then the cells are going to complete the cycle and divide. Again, of the choices you were given, that's the best one. Obvi, all these other checkpoints have to be made, but of the choices you were given, that's the best choice. All right, and then on 7, 8, 9, and 10, I'm going to explain these by kind of just going over what's happening in the diagram. So let's look at our axes labels. This is the amount of DNA in the nucleus. And here we are shown the first and second cycles of cell division. So this is your starting amount of DNA. And then you can see here in panel 2, it's increasing. It's actually doubled. So I can infer that panel 2 is the S portion of interphase, which means that this has to be G1. Then the DNA stays doubled for a little while, implying that this has to be G2, and these are the final preparations for mitosis. Then the amount of DNA starts to decrease because the nucleus is being divided. The chromosomes are being the chromosomes and the copies are being separated, which then puts us down to the end amount of DNA, which is the same as what we started with in the 2 new cells. And so that means that 1 is G1, this is S, this is G2, IV4 is mitosis, and then these are the new cells in G1.